This is part 3 of the Chapter 35 lecture notes, and this is where we left off. By the way, the previous slide had, well, I'll just show you. Okay, so I tried to click on where it says figure and figure, and I'm not sure what the problem was, but it's a publisher problem, so I don't know what figures were supposed to be there. But I think this figure is sufficient, kind of explains things. Um, an action potential or a nerve impulse is an electrical impulse that travels from this point, which is called, or somewhere around here anyway, where the axon begins is called the axon hillock, just where the axon begins. That the nerve impulse travels from this point all the way down the axon until it reaches what's called the ends of the axon are called the axon terminals. So each one of these little ends are axon terminals, and they stimulate different areas of the target or different targets. So if you look at picture A, it says in response to a signal, and the signal is going to come from the dendrites or the cell body, or it could be a combination of signals coming in from several dendrites and several areas on the cell body. And but that signal has to be strong enough to cause depolarization at the axon hillock um, to threshold, like we talked about. It has to cause the voltage at the axon hillock to go from -70 to at least -55. And so, in response to the signal, the soma end of the axon, which we call the axon hillock, becomes depolarized. So, it's going from -70 into sorry, that didn't right, -70 millivolts toward a more positive direction. Um, the the inside of the plasma membrane started out negative and it's becoming more positive. So that's what depolarization means. And the depolarization spreads down the axon. And then it says, meanwhile, the first part of the membrane repolarizes because sodium channels are becoming inactivated. Okay, so to explain B, what happens is there's a beginning that causes the nerve impulse, um, that results in a nerve impulse traveling down the axon and sending a signal. But then you don't want a nerve impulse to continue indefinitely. You want there to be an end. So after depolarization, then you have to have repolarization. So what happens, the membrane has to repolarize back to what it was in the beginning, which is -70. So what's going to cause that is the sodium gates are going to inactivate. In other words, sodium can't come in anymore. Sodium can't come into the plasma membrane anymore. What was causing the depolarization was sodium ions moving into the plasma membrane. And what's going to cause repolarization is inactivating the sodium channels, so sodium can no longer come in, and and then the opening of potassium channels, because potassium goes in the opposite direction. Potassium will actually leave. So then instead of having positive charges entering the cell, you have positive charges leaving, making the inside more negative once again. And then in part C, once this is over and once the membrane potential reaches -70, the membrane potential returns to resting. And but what you can see happening is the depolarization travels. It's traveling from all the way down the axon. It's just traveling in this direction. but as it travels, the part that already was depolarized becomes repolarized and then returns to resting. And you end up with the entire axon returning to the resting membrane potential when it's all over with. Um, so different things will cause voltage-gated uh, channels to open and an action potential to be generated. Um, a chemical synapse is one that is controlled by a chemical called a neurotransmitter. So at a chemical synapse, this is what happens. Depolarization um, causes voltage-gated calcium channels to open. Calcium, of course, is another cation or positive ion. Calcium ions will initiate synaptic vesicles to fuse with the presynaptic membrane. That means the membrane in the um, I guess the first neuron. The synaptic vesicles will fuse with the presynaptic membrane, actually the axon terminals of the presynaptic membrane. And then they will release their neurotransmitters into the synaptic cleft, which is the gap between the axon terminals of the presynaptic neuron and the um, target, which could be another neuron. So neurotransmitters such as acetylcholine or serotonin or norepinephrine is released into the synaptic cleft, and then that neurotransmitter um, will attach to the postsynaptic membrane to a receptor on the postsynaptic membrane, and it will cause the postsynaptic membrane to respond. And what you can see, in fact, let me blow this up a little bit because it's even small for pointer options. Okay, so starting with step one. So what you're looking at is this, okay, you'll follow the hand. You're looking at the axon of a presynaptic neuron. That means the neuron that's sending the nerve impulse to a second neuron. So the first neuron is called the presynaptic neuron. And here's the axon, and then these are the axon terminals. And you're looking at the area where the axon terminal of the presynaptic neuron is coming in contact with the cell body or the soma of the postsynaptic neuron. So here's your presynaptic neuron, and your action potential is arriving at the axon terminal. The action potential or the nerve impulse is arriving at the axon terminal. And that is going to cause voltage gated calcium channels to open, and calcium ions will enter the axon terminal and lead to these vesicles, which are called synaptic vesicles, to fuse with the membrane of the presynaptic neuron, the membrane of the axon terminal of the presynaptic neuron. And once they fuse, they release their neurotransmitter by exocytosis. And then the neurotransmitter will enter the synaptic cleft, which is the gap in between the pre and postsynaptic neurons, and it will bind to receptors on the postsynaptic neuron, on the membrane of the postsynaptic neuron. And then that receptor is attached to a gated channel, and that channel will open, and sodium will come in, or some ion will come in, and change the voltage across the membrane of that postsynaptic neuron. And you can see that here. You can see the neurotransmitter is binding, the channel is opening, and the channel would open and some ion would move in, and maybe out. But anyway, some change would occur in the resting membrane potential of the postsynaptic neuron. And then eventually the neurotransmitter would be, trying to think of how to say this. What you would have is reuptake of the neurotransmitter into the presynaptic neuron, into the axon terminal, because again, you can't have neurotransmitter acting indefinitely. There has to be a stopping point. Okay, now we're going back to structure. In the central nervous system, um, which is the brain and the spinal cord, the membranes that wrap around the brain and spinal cord are called the meninges. You've probably heard of meningitis before. And the meninges are the um, dura mater, the arachnoid mater, and the pia mater. So this is the dura mater here, and then the arachnoid mater here, and the pia mater is here. So that's the order. The spaces between the meninges are filled with cerebrospinal fluid, or CSF, which are produced by ependymal cells. I don't know if you remember, ependymal cells are the cells, the neuroglial cells that produce cerebrospinal fluid. And the fluid is continuous with the 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 fluid in the spinal cord and the ventricles of the brain. All right, if we look at the structure of a typical spinal cord, a cross section through it, I'm sure you've heard the terms white matter and gray matter. Gray matter actually gets its color from Nissl bodies. Nissl bodies are just groups of ribosomes and rough endoplasmic reticulum, um, and those are found in the cell body, or the soma of the neuron. So gray matter gets its gray color from those Nissl bodies. Um, but the point is, if the Nissl bodies are found in the cell body, in the soma, then gray matter contains a lot of cell bodies of neurons. And white matter contains myelinated axons. And those myelinated axons cause the white color, because myelinated axons are, the myelin is filled with fat, or lipids, which makes it look white. So the spinal cord has two main functions. The functions are reflex actions and communication. Um, with the brain and the spinal nerves. And the spinal cord, just like the brain, is composed of gray and white matter. The gray matter is filled with cell bodies of neurons and unmyelinated axons. So the cell bodies and the dendrites and the unmyelinated axons are what you find in gray matter. And then myelinated axons are what you find in white matter. All right, here you see another cross section showing a reflex arc. So a really good example for everyone to relate to is you get um, pierced by a pin in your skin, whether you step on it or just stick it in yourself by accident. So receptors in your skin detect that pain and send a signal to the spinal cord through the dorsal root ganglion, which is just the dorsal side of the spinal cord. And so interneurons within the spinal cord integrate that information and send a signal to your skeletal muscle to cause you to move your um, skin away from the pain, away from the source of pain. And that's the um, ventral root of the spinal cord.